Good morning, everybody. Welcome to a very special 25th anniversary of CMV MBS Research Day. I will let Dr. Vanderwood tell you the special name that she has coined for this celebration. But just have a couple of quick announcements. This is really a special event. We have several new things that we've never done for Research Day before. Last night we had an excellent invited speaker, Dr. Cheryl London from Tufts, that gave a great talk. We celebrated the past 20 Zoetis/Pfizer Research Day Award winners at the TMI, had a really nice social, had a great turnout. And now today for the main event, we have our keynote lecture from this year's Zoetis Award winner. Dean Vanderwood will introduce that speaker shortly. We also have, if you look around you up in the balcony, our first, what will be annual undergraduate research symposium as part of CVMBS Research Day. We've got 36 undergraduates presenting their work today, which we're very excited about. And then finally, at the end of the day, we will be giving out our annual Golden Pipette Award to the department with the highest composite score as determined by a secret proprietary algorithm that I am not privy to. But it is equitable, I have been assured. And then really excited, just before the Golden Pipette, we will award our inaugural Green Pipette Award. This is a new award envisioned by Tracy Webb and Colleen Duncan to award the department that demonstrates a commitment to sustainability and efficiency in our laboratory spaces. So really excited to award that this afternoon. I encourage you guys all to stay, if you can, for that award ceremony. It should be really, really fun. Just wanted to thank our sponsors real quick for the overall Research Day. Boehringer Ingelheim, Zoetis, Lisa Bergeron will be presenting the Zoetis Award to Lynn after Lynn's talk, and CSU Strata. I have a message here from Dr. Mike Lappin, who could not be with us here today. I have a bit of an unpredictable cough here. So Mike sends this message. So on behalf of Dr. Mike Lappin and the Center for Companion Animal Studies Young Investigator Award Program, we thank Purina, Antec, Merck, Vetoquinol, Elanco, Zoetis, and Boehringer Ingelheim as core sponsors of the Young Investigator Award this year. Thanks for your continued support. And I feel like I'm stealing Mike's thunder a little bit here, but I can't hold this in. In addition, Purina just gifted a $1.5 million gift to the Center for Companion Animal Studies Young Investigator Award Program to endow it in perpetuity. And that is a big deal for Mike and for our college. So thank you, Purina. And with that, so after the Q&A, we're going to have the award presentation, a few pictures, and then immediately following, we're going to have our undergraduate research symposium. We have some food and drink available, so we encourage you to stay here. And then after that, we're going to migrate over to the main Laurie Student Center where we have our main event, which should be familiar to many of you, okay? And with that, it's my great pleasure to bring up Dean Vanderwood.
Thanks so much, Mark, and thanks to Mark and his staff and the committee for Research Day for putting together a really fantastic event for what we should be calling the Silver Jubilee, the 25th anniversary. I just finished watching the 6th year of the Crown series, so I'm really into the nomenclature for that type of recognition. This event is really near and dear to my heart, an event that micromanaged from 2003 to 2007 when I was the Phi Zeta faculty advisor, and that was how this event started. As Dr. Mama pointed out last night at the opening ceremonies, it was really a Phi Zeta Honor Society that started this event, and it was student-driven. Then from about 2011 to 2019, I was Associate Dean for Research and had this very fantastic committee to put together this event and continue it and to grow it. And I'm so excited that this year we have the biggest, by far, number of abstracts submitted, almost 200. And this is undergraduate, veterinary students, graduate students, veterinary researchers, postdocs, a few staff, but almost exclusively our student body and our young researchers that are representing all four of our departments, the very broad research that we do here, and we can do because our faculty are extraordinarily productive and bring in almost $50 to $60 million in extramural support every year to support this. So I'm really proud of this event. As you all know, probably I'm a big research nerd, and this is one of my favorite events that we have every year in the college. I would like to introduce two other special guests joining us today who are sitting in the shadows but in the front. Our new provost, Dr. Marion Underwood, and our new VPR, Dr. Cass Mosley. Would you both stand up? And Dr. Underwood started just about a month ago. She joined us from Purdue. Her background is in childhood behavior, and Dr. Mosley joined us from University of Oregon, and her background is in forestry and ecology. So I'm sure they'll see some things on the slides that are interactive with they're not, or in our posters today. And I'm just really excited to have them here so we can show off all of our wonderful students and all the amazing work that they're doing. So as Mark alluded to, CVMBS Research Day has traditionally been kicked off by a keynote speech by the most recent recipient of what is now called the Zoetis Award for Veterinary Research Excellence. I have to say it has had different names as different companies have kind of changed hands, but it has been in effect for about 20 years. This is a competitive award selected from our assistant professor and early associate professors in our college who show extraordinary research productivity and early career success. And as part of the Silver Jubilee, as Mark noted, we are welcoming back former recipients of the award. We have special t-shirts that are printed up that has everybody's name. And I think we had 18 individuals who are going to be with us part of the day that are part of that list. And they're really some of the faculty that drive a lot of the research going on here and are mentors of the students that will be presenting today. I'd like to ask everybody in the audience who is part of that incredible cohort to please stand up, if you would, and we'll give you a round of applause for our alumni Zoetis Researcher of the Award winners. Thanks very much. So it's with a lot of excitement and enthusiasm that I share with you this year's awardee, Dr. Lynn Pezzanite. Dr. Pezzanite came to CSU to do her equine surgery residency in 2015, completed her residency and master's degree in 2018, and then went on and completed her PhD in 2021. Now remember, that's 3 years ago, right? She participated when she was doing her fellowship in the Colorado Clinical Translational Science Institute NIH-supported fellowship. That's a collaborative award with the University of Colorado Denver, and then she received a position on the CVMBS T32 NIH Postdoctoral Fellowship Award. She's also received the prestigious and highly competitive CCTSI Co-Pilot Award, funds from the Grayson Jockey Club. She has an NIH Loan Repayment Award, and she has other work sponsored by industry, et cetera. And she already lists, remember, she got her PhD in 2021, 36 peer-reviewed publications, and another 12 in preparation or in review, which is just phenomenal to be that productive at this point in her very young career. She's also presented 70 posters or oral presentations at local, national, international venues. She's produced 8 book chapters and holds 3 patents. She already has many awards, as you can imagine, from that portfolio, including recently being named the American Veterinary Medical Association and Foundation Early Stage Investigator Research Awardee, which is a national award to early career investigators. She is a Wiley top-cited author, and she's the recipient of the University of Colorado Orthopedic Research Symposium Poster Award and many other things that I didn't want to take up all her time listing her awards. She conducted her undergraduate work at Purdue, so you can talk to Dr. Underwood about your experiences there. She was named the Outstanding Senior in Animal Science when she graduated from Purdue, and she obtained her DVM degree from Cornell, and not surprisingly, has many awards from that experience as well, including the American College of Veterinary Surgeon Large Animal Surgery Award. And with great excitement, I'd like to also announce that Dr. Pezzanite has just accepted a tenure-track faculty line at the Orthopedic Research Center as a cellular and molecular biologist. So she'll be officially starting her job as an assistant professor next Thursday on February 1st. Really exciting for us. So we can congratulate ourselves on recruiting such fine talent, and also congratulate and thank Dr. Pezzanite and welcome her to the podium to present her research, after which we'll give her award. So Lynn, please come up here and thank you.
All right, well, it's my honor to be up here today, but what I really want to emphasize is that today is really about my mentors, recognizing my mentors and acknowledging my collaborators that have helped me get to this point in my career. The research I'll talk about today involves investigators from 4 different veterinary schools, primary collaborators at the University of Colorado Department of Orthopedics, as well as multiple other departments on campus. As Dr. Vanderwood said, I'm a veterinary surgeon. My research program focuses primarily in comparative orthopedics and musculoskeletal regeneration. And what I wanted to do today was frame how my mentorship throughout my career has really gotten me to this stage. So beginning at the very beginning, why orthopedics? So my family moved between the US and Europe throughout my childhood for my dad's position, but I went to high school in Middlebury, Indiana, which is a small town in northern Indiana. I played a lot of sports growing up, was best at track. My junior year of high school, I was captain of the state champion track team, but went on to, 3 of the 4 of us went on to develop injuries going into my senior year. And at the time I was being recruited by several Division I schools, thought I was going to pursue my track career in college and then beyond that. Wasn't really thinking about academic pursuits at the time. Identifying as an athlete was really my primary focus in that moment. So going into senior year and not being able to compete was a very big deal at the time for me. Coach Carson was my primary mentor. He had been a very good baseball player. His mentorship style resembled more, and pep talks, the picture on the right, generally ending in some sort of baseball metaphor. His favorite movie was Bull Durham, and so when I was going through this, trying to figure out what I was going to do, didn't think I was going to come back to compete at the same level, if at all, my senior year of high school, and then knowing my Division I dreams were not going to happen. He was like, look, Pezzanite, that was how I remember our conversations always starting. Look, Pezzanite, get over it. Stop feeling sorry for yourself. It's like in Bull Durham. You should just be happy to be here and try to help the ball club. And that is how I approached my senior year as captain. We did go on to get second at the state championship my senior year as well. So at that point, I really had to shift directions from what I thought I was going to do. My mom was a veterinarian, and so in terms of my career goals, it was between veterinary school and being an architect at that point was my backup plan. Purdue University was my in-state veterinary college, and they offered me an academic scholarship in the Department of Animal Sciences. So I went to undergrad at Purdue. I had the opportunity to work for these 6 wonderful men who are all still there. Barry Delks, Mike Neary, Scott Mills, Mark Russell, who's now the Dean of Agriculture, John Patterson, and Scott Donkin. I worked for each of them either in a research capacity or as a teaching assistant over the course of those 4 years. But I wanted to highlight Barry, who was my academic advisor, was one of the first people that I remember believing that I could go to vet school and go to whatever vet school I wanted to. The quote I always associate with Barry was, leading through service, being a servant leader, and putting other people's needs before your own. When I think of him, I always remember how much power each of us has to have a positive impact on another person's career. And so I did end up getting into 4 vet schools, elected at that time to go to Cornell, mainly after discussing it with him and for various reasons. Moved to New York, didn't know anyone in New York at the time, and I needed a job, any job. And Lisa Fortier, who was still is, equine orthopedic surgeon, spoke in one of our classes, and she had just gotten a big grant, needed to staff up her lab, and so I asked if I could work for her after that. Started working in her lab early in my first year of vet school. To highlight the program that they had created really at the time in comparative orthopedics, she and Alan Nixon had really built up that research program. There were, and this is not a comprehensive list, but just in the time I was there, they trained 4 boarded surgeons in PhDs, Lauren Schnabel, Kyla Ortvedt, Heidi Riesink, and Michelle Delco, who have since been known as the Golden Girls. Each went on to run their own academic programs as faculty at Cornell, North Carolina, and UPenn. But I had the opportunity to work most closely with Lauren Schnabel, who's in the third picture, and then in the right again with Lisa, closely during her PhD. So I got started in the lab, was doing things like dishwashing, that sort of thing, and she really kind of took me under her wing. I got involved with their version of the Vet Summer Scholars Program, which we called the Veterinary Investigator Program, or VIP, and then worked closely with her when she was under the mentorship of Lisa and Doug Ansack in the picture in the middle, who's an immunologist at the Baker Institute at Cornell. And so this was really kind of the first example in my career of interdisciplinary teams working together. Lisa had started a program they called Cartilage Journal Club, which involved immunologists, engineers, a bunch of different specialties beyond just the clinical group. And so that was a really important first example of team science to me, as well as having these really strong female surgery mentors early in my career, I think was really pivotal. And I had done some research, some data collection, again, general lab work in undergrad, but getting involved with that group, I was on my first authorship and second authorship working on Lauren Schnabel's PhD work. So that was really important moving forward. Up to that point in my career, I had thought I was likely going to do mixed animal practice, like my mom had, and so this was really kind of the first time when I thought I was really going to pursue a surgical residency. So moving forward, clinically, I did, after that point, I pursued an internship in equine practice at Littleton Equine Medical Center, which is just down in Littleton, Colorado, and then matched a surgical residency here, which was my first choice, really based on the reputation of the orthopedic research that had come out of here up to that point. I wanted to highlight at this point my intern mates and resident mates. I had the privilege of working with some amazing surgeons during this clinical training period. Lori Goodrich, Dean Hendrickson, Chris Kowchek, Wayne McElraith, the list goes on. Was able to get involved with Jeremiah Easley in the preclinical surgery research lab, and I remember him saying this for a long time: if you want to go far, go together. And I would not have made it through that period of time without this amazing group of individuals that I worked with. Sometimes residency looks like this in the middle, and sometimes it just looks like this. And you know, having those teammates around you was just so important during that period of time. To highlight just some of the other clinical opportunities I had, I think this was a really pivotal time for me. These are opportunities, these were all patients that I had the opportunity to work with Dr. Hendrickson at various times, and have gone on to do a fellowship training under his supervision in minimally invasive surgery since then. But I think even though I've moved into a more research-based role, having this basis of surgical skills is really important for the preclinical modeling work that I'm now focusing on. So brings me to my PhD training. So after residency at Colorado State, stayed on in a PhD program under the supervision of Steve Dow and Lori Goodrich. And I knew at that point that I wanted to focus on improving outcomes in musculoskeletal trauma. That was my main focus, my main interest after my residency, particularly infected and chronically non-healing tissues. And I think this was my PhD mentor team. I had such a great group around me during that time. Steve Dow is an internal medicine clinician and immunologist, Lori and Dean Hendrickson as clinical mentors, Kelly Santangelo from MIP, and then in the middle, Jason Stoneback, who's an MD, Chief of Orthopedic Trauma and Fracture Surgery at University of Colorado, Denver. And so I think one of the best opportunities that I have ever had, honestly, is having a year of my PhD funded through the CCTSI, as Dr. Vanderoo mentioned. So this program, you basically have 2 mentors, one at your home campus, and then one translational mentor in your area to increase crosstalk between campuses and then just increase the amount of communication between clinicians and basic researchers. So Jason Stoneback was my translational mentor for that program. One fact that some people know about Jason is that he was a professional bull rider, made his way through med school that way, but also still owns horses himself, is still part of the rodeo world. He was just volunteering his time with the stock show as the clinician for the rodeo cowboys there. So we had a lot in common just in terms of our backgrounds, and I think this team and being built in this way with him as both a clinical and research mentor and then the interdisciplinary team, I think really embodies this quote that Steve always says, science is a team sport, and I think Dr. Dow does that better than anyone I've ever met. So that was the basis for my PhD committee and work moving forward. So in the theme of trying to improve outcomes in infected and chronically non-healing tissues, previous work from Dr. Dow's lab has looked at this concept, basically with the underlying premise that the increasing incidence of antimicrobial resistance in both human and veterinary medicine necessitates development of novel therapies to treat infection. And they and other groups have shown that cellular therapies have these inherent antimicrobial and immunomodulatory properties through peptide secretion and paracrine recruitment of immune effector cells, and that these properties could be enhanced through pre-activation of MSCs with various toll and nod-like receptors that are found generally in inflammation and infection. So just showing some of his prelim data, this is not mine, but just generally from the lab, that this pre-activation with toll-like receptors such as TLR3, poly-IC, enhanced neutrophil recruitment and engulfment of bacteria, altered macrophage polarization in treated tissues, and then enhanced resolution of infection in mice compared to non-activated or antibiotics alone. So we wanted to build on this, and working with Dr. Stoneback's group as well. The program he runs there, called the Limb Restoration Program at University of Colorado, Denver, treats non-union fractures, osseointegrated patients, patients with significant bone loss in fracture healing, most of which are complicated by infection. And so his interest was also in pursuing this as an adjunctive therapy to standard antimicrobial therapy, which is not effective in some cases, necessitating amputation. So we wanted to move this into a large animal model of infection, and so we looked at it in the equine model. This was basically a MRSA, MDR MRSA strain, USA 300, of septic arthritis. The horses received either standard antibiotic care or antibiotics and activated stromal cell therapy. And we did demonstrate at 2 weeks significant improvement in their clinical pain scoring, bacterial bioburden in both synovium and synovial fluid in those treated joints, and then evidence of immunomodulation, both systemically in their cytokine profiles in plasma as well as in synovial fluid. So just showing some of that data, significant reductions in pro-inflammatory IL-6 and IL-18. And you can see, even just grossly, the difference in the synovial fluid normalization in the stem cell treated horses. More recently, we did a targeted transcriptomic analysis to evaluate this further and basically demonstrated mechanistically that the activated stromal cells are inducing a more T-regulatory response in those treated joints compared to untreated, where there is a higher profile of innate immune gene expression. So currently, since I've transitioned from PhD to a faculty role, we're looking at this moving towards ideally phase 1 clinical trials in humans in Dr. Stoneback's limb restoration program. We're also pursuing further funding through federal funding sources to look at this in large animal models, particularly in the context of bone-anchored limbs. So in the image on the lower right, you can see this individual with an osseointegrated prosthesis. Dr. Stoneback is one of the only surgeons in the country and world who perform bone-anchored limb surgical prostheses procedures. And so at the residual limb interface, infection is a huge problem, which in some instances necessitates removal of the prosthesis, and that's our goal for integrating this activated cellular therapy in that context as well. So moving kind of into my current research program, in conversations, again, with the limb restoration team, many of these post-trauma individuals also develop post-traumatic osteoarthritis, and that's a very common condition for us to treat in horses. It is the most common joint disorder in horses, important source of reduced athleticism and economic loss. It is a heterogeneous condition just as it is in people, which is associated with obesity and aging. The prevalence really varies depending on which source you look at. Up to a third of young athletic thoroughbreds have been shown to develop PTOA, and some sources report 80% of horses over 15 years. So spontaneous OA in horses is certainly common for us, and has translational relevance to humans also suffering from PTOA processes. So a number of papers out of our group and Lisa Fortier's group in New York have talked about this. The equine model is a good one for human PTOA due to their similarity in joint volume, cartilage thickness, and loading force. The large joint volume also allows for repeated collection of synovial fluid for analyses with more parameters compared to other laboratory species or models that we have. And as we've already said, OA and septic arthritis are common naturally occurring processes. In terms of rationale as well for further studies, a number of these recent papers have recognized a need for a consensus on several factors associated with OA, so nomenclature defining specific OA disease processes, identification of biomarkers that predict progression, or to refine the taxonomic classifications, as well as identify individuals that are at greater risk following a traumatic event involving an articular structure, monitoring OA development in response to therapy, and predicting long-term clinical outcomes. So we were recently funded as a team, so myself, Lori Goodrich, Steve Dow, and Jason Stoneback by the Grayson Jockey Club to use single-cell RNA sequencing techniques to evaluate temporal progression of osteoarthritis. So again, focusing on that really early stages of progression and using these transcriptomic techniques that have only fairly recently become available with the goal of defining that early stage of progression and then long-term having gene therapeutic or biologic interventions that are tailored more to disease stage. So to demonstrate a little bit of our initial work, we have done this in 3 clinical cases. So not the induced model, but horses with naturally occurring disease. And I'm not going to go, most of what I'm going to present today, my students are presenting in greater detail later, so as not to steal their thunder, it's going to be somewhat of a broad overview. But essentially, what we've seen initially is similar to what has been reported in some of the recent human papers. Single-cell RNA sequencing revealed 8 major cell populations in equine synovial fluid. These are horses with late-stage degenerative joint disease. Of those, when we look at the number of differential genes expressed, macrophages, besides cycling cells, macrophages had the highest number of differential gene expression. When we present the data by healthy versus OA, so healthy in green and OA in purple in the graph on the left, we found an interesting trend that macrophages, again, and dendritic cell clusters were largely coming from the OA synovial fluid samples, while both the CD4 and CD8 T cells were coming from the normal synovial fluid samples, indicating a shift in the immune composition of synovial fluid following onset of OA. Highlighting, when we've dug into this data a little bit further, specifically myeloid cells and IL-23 gamma delta T cells were overrepresented, which is something we're looking at further. When we pull out just the myeloid cell population, we saw 12 distinct myeloid cell types were identified with independent reclustering. And then when we show here the differential gene expression, 47 genes were highly expressed in OA and 66 in healthy tissues, depicted in the volcano plot on the left. Overall, 84 pathways were enriched in OA specific to myeloid cell lines, highlighting a few of these related to cell migration, chemotaxis, and lymphocyte activation. Again, ultimately suggestive that OA myeloid cells play a role in chemotaxis and migration, as well as immune cell activation in OA processes using the relevant equine model. So, impact of this work generally highlights the complexity of myeloid cell populations associated with OA progression that wouldn't be possible using other methods besides single-cell sequencing. These findings support macrophage interactions and IL-23 T cells as important contributors to disease, supporting further investigation of therapies targeting these IL-17 and IL-23 inhibitory pathways. And again, moving forward, we are funded to look at this temporally in early stages of OA using single-cell RNA sequencing. We did operate those horses in November and December, and this is the whole team, including the UC Denver group that came down to help with those surgeries. So as we've been performing these single-cell sequencing studies kind of concurrently, we began to think more globally about OA as a systemic inflammatory process. During that period of time, Dr. Stomack introduced me to Mike Zusick, who's a professor of orthopedics, also in the Department of Orthopedics at UC Denver. And he has a great program focusing primarily on osteoarthritis and microbiome dysregulation, perpetuating musculoskeletal conditions. So in this study, we wanted to compare, just using simple bulk RNA sequencing techniques, the transcriptomic responses in synovial fluid cells and then correlating those with peripheral blood mononuclear cells in circulation, as well as systemic inflammatory cytokine profiles using this expanded biomarker panel, which hasn't previously been integrated in this fashion. So what we showed initially was these are synovial fluid cells, again, significant differential gene expression in horses with naturally occurring osteoarthritis, over 800 genes upregulated, 350 downregulated, and that correlated with increases in a number of synovial fluid cytokines, again highlighting IL-17 here, which was also seen in our single-cell sequencing data, as well as a number of other known ones, IL-1 beta, IL-6, IL-18, IP-10, and GCSF. But perhaps more interesting was that we also saw differences in transcriptome of the circulating peripheral blood mononuclear cells. So 41 upregulated genes and 55 downregulated, and similar profiles in plasma in these same horses, so sampling the same cell population. When we look at gene expression pathways that were related to cellular functions that were upregulated in both, we did identify several that were, generally they were related to cell metabolism and motility, ECM, proteoglycan generation, insulin uptake, and ATP binding. So this work, to this point, has enhanced our mechanistic understanding of the cellular and molecular underpinnings of synovial immune pathways. We've identified several new local and systemic cellular biomarkers that may be associated in future studies with clinical and imaging observations. One key finding that I didn't mention was one of the most upregulated genes was related to a serotonin receptor. Serotonin receptor antagonism has not been explored in equine OA, but this represents one potential avenue for us to treat chronic pain associated with OA. And then moving forward, we'd like to also, which we're doing in the Grayson-funded study, look longitudinally to evaluate transcriptomic alterations in circulating leukocytes and correlate that with the changes we're seeing locally in the joint. Building on this work a little further, and again with the emphasis on systemic inflammation exacerbating OA rather than joint-centric approaches that we've perhaps typically taken, at least in equine surgery. One major focus of my lab moving forward, again working with Dr. Mike Zusick, these are a couple of recent review articles he's put out on this topic, is exploring this concept of the gut microbiome joint connection, what implications that has, particularly in the osteoarthritis of obesity. So obesity is common in our riding horses as well. Up to 20 to 45% of pet horses in the US are considered obese. So again, and many of those have OA in multiple joints, as humans have been reported to have as well. So another example where we can take a transdisciplinary approach, working in our species of interest to potentially inform human outcomes as well. So we just took an initial look, and again, I'm going to present this at a very high level because I want you all to come to Zoe Williams' talk later today to hear more about it. But generally, we just wanted to screen horses with OA, again, naturally occurring degenerative joint disease, with and without OA, to see if we saw similar microbiome changes as have been reported in some laboratory species of induced OA. And we saw significant differences, as well as an overall reduction in microbial diversity. If in general we think of diversity as a good thing, that is reduced in OA disease states. And then an alteration in relative abundance of bacterial phyla with an increase in firmicutes in the OA cases, which is similar to what they've seen with people with naturally occurring disease. We looked further at, again, taking these same leukocytes from horses with OA versus not, and we demonstrated that those circulating leukocytes also contained microbial DNA, which was distinctly grouped between OA and healthy horses, and that the distribution of that microbial phyla resembled that we saw in feces of horses with OA, indicating that potentially microbial translocation from the gut to systemic circulation may be one route by which dysregulation of the microbiome in the OA of obesity is exacerbating inflammation at distant sites such as the joint. We evaluated this a bit further, looking at this interactome, and we further demonstrated that specific taxa are correlated to transcriptomic differences in OA versus healthy horses. Again, primarily implicating firmicutes as one of the most important phyla, but generally these transcriptomic responses were related to cell structure and the innate immune response. So what is generally the impact of this work? We showed here that circulating leukocytes from horses with OA contain bacterial DNA, which has not been previously reported. The microbiome sequence from those PBMCs resembled that in feces from OA versus healthy horses. The presence of specific microbiota taxa correlated to genome of the OA versus healthy horses. And this presents several future avenues for treatments directed towards microbiome dysregulation in multi-joint OA and systemic inflammation overall versus, again, the joint-centric approaches we've historically taken. So again, these data are really preliminary, raise a lot of questions and avenues for future directions. Are the bacteria that have previously been reported in joints actually alive or just DNA remnants? How the microorganism DNA is transported to the joint? And generally longitudinally examining alterations within all three cell populations, so feces, circulation, and then in joints. So this concept is the basis of my current KO1 proposal with Dr. Zusick as my primary mentor. And then I wanted to end with just really briefly touching on a few things our lab is working on, generally related to musculoskeletal disease, but more interventionally oriented. So several topics, looking further at the MSC activation techniques, not just for septic arthritis, but joint disease in general, OA processes, immunotherapy techniques for tendon disease, local approaches in fractures, and then xenotherapeutics. So very briefly, we've extrapolated some of the work that was previously done in treatment of infection for treatment of OA. This is demonstrating some of that, showing improvement in mice with induced PTOA and a number of gait parameters, as well as differential gene expression from those joints following 2 doses of activated mesenchymal stromal cell therapy. So we're currently repeating this study to look at that further. We have, working with Kirk McGilvray in the OBRL, we've applied some of these immune agonist techniques to rotator cuff tendon injuries, working in rat models, using melt electrowriting approaches to 3D printed biocompatible implants, incorporating the same agonists we've explored in other avenues. Some initial really interesting activity data indicate that rats treated with specific agonists return to activity sooner, which may have important implications for early recovery for athletes. Working with Nick Alfonso, who's also a member of the Trauma Fracture Surgery Service at UC Denver, as well as Kirk McGilvray and Steve Dow, we're incorporating, again, local techniques for improved treatment of open fracture wounds. Generally, one issue besides multi-drug resistance, but just generally open fractures still maintain a high infection rate, up to 40%, depending on the grade. And so their group is interested in field deployable dressings for acute wound management. So that's been another great interdisciplinary collaboration that's kind of just getting started. And then finally, one area I'm really interested in doing more work in, and we have one initial grant looking at this, is selective clearance of senescent cells, which have been shown to exacerbate multiple disease processes associated with aging, including osteoarthritis and potentially delayed fracture healing in elderly individuals. So we're just beginning these studies, but the long-term goal is improved treatment of OA, at least initially with xenotherapeutic strategies. So generally, summarizing that area in our lab is we think the available cellular-based therapies can be enhanced for multiple MSK applications for improved immunomodulation and antimicrobial action. Potentially, our current strategies for open fracture wound management may be refined with these local therapies. And in multiple contexts, musculoskeletal tissues undergo senescence, and selective clearance of those senolytic cells may be an avenue for treatment. So generally, moving into this position, I'm excited to apply a number of these cellular and molecular techniques, leveraging my surgical training and development of preclinical models, specifically for musculoskeletal disease, and then I'm really excited to continue these collaborations, both across our departments here at Colorado State and then also with UCAMC. And then I wanted to end really with my students, because that's why we're here today. I've had the opportunity to work with an amazing group of students, many of which have wanted to go on to surgical residencies. Gabby and Aya were 2 of my first during my PhD. Gabby went and did an equine internship, and then we were able to bring her back as a surgical resident. Robert was an intern, matched a surgical residency in small animal. Aaron Webster was also a Vet Summer Scholars student, is currently in an internship at Cornell and applying for surgical residencies. And Megan and Blaine, we're still lucky to have here with us, both former Vet Summer Scholars students and presenting today as well, and both will be in the match for surgical residencies here soon. I've had the opportunity to do mentoring in a number of different areas, but I think what I always remember is the opportunities given to me, and I always think of that when a new student comes and wants to work in the lab. This was me 10 years ago with Lauren Schnabel at my Vet Summer Scholars Project, and me last year with Blaine and Megan at theirs in Minnesota. So generally, future directions, I'm really invested in creating additional learning opportunities for our residents, veterinary and graduate students, mainly by emphasizing comparative approaches across species and further developing these relationships with human specialists, generally with a commitment to advancing animal and human welfare through a comparative orthopedic approach. And I just wanted to end with my take-home messages from my mentors. It's a privilege to do what we do for a living, so be grateful. I'm just happy to be here. Lead through service. Every day is a school day. Science is a team sport, and if you want to go far, go together. And I'll end with, there's so many people that contributed to the work I presented today, but I really want to acknowledge Linda Chow and Dylan Amons, who helped with a lot of the sequencing work. And then I hope you can all see my students' projects today, Megan, Ashna, Cody, and Zoe. I hope you have the opportunity to see their presentations later, and I'd be happy to take any questions.
Great talk. Great breadth. Great detail. I love how you bring molecular and cellular state-of-the-art techniques to veterinary medicine. It's really impressive. And I really appreciate centering this whole thing around your students. It's really appropriate for today. Thank you. This is open for questions. So discussions, comments, questions. I invite graduate students or undergraduate students to ask the first question.
Hi. Oh, God. That was so cool. I was just wondering about the practicality of your fecal analysis, if you thought it was practical to implement it for early OA diagnostics, like in a clinical setting.
Yeah, that's a really interesting question. We haven't really approached it from a diagnostic standpoint. I actually haven't thought about it from that perspective, but yeah, I think as a long-term goal, though, I think that would be really useful in terms of knowing, like, should we be, that's kind of our goal with the transcriptomic work in general, is what is that tipping point in OA where we should be intervening, and can we intervene before that? So I think coupling that information, which we will have from this current ongoing study where we're sampling the joint as well as the circulating leukocytes and feces at once, I think having that correlative information, maybe we may be able to answer that question. But great question, yeah.
Thank you.
Do you foresee any of your treatment options that you've found effective for osteoarthritis being applicable to other forms of arthritis, like rheumatoid or other factors?
Yeah, it's a good question. We don't, I guess working in the equine model, we don't really deal with rheumatoid. And in general, I think the transcriptomic data have suggested that they may be very different. So it's not really something we're currently interested in, but certainly I think could be potentially applicable in the future, yeah.
Hi, I am very interested in the hydrogel that you are working on. Will you talk a bit more about, is that going to be implemented right when the injury occurs, or is that going to be after surgery, or something like that?
Yeah, our goal with the hydrogels is really to have a general hydrogel base that promotes wound healing and then be able to vary the components to address basically different stages of healing, like you suggested. So there would be one composition potentially that could be applied by first responders, and then another after the surgeon performs his lavage and debridement that he would place in the wound prior to primary closure. So ideally it would be variable, yeah.
Great talk, Lynn. I'm glad to see gamma delta T cells on the list, as you know. What poster or what talk is going to have the gamma delta T cells? I didn't catch that. And I wanted to hear a little bit more, because you briefly said There was a subset.
Dylan Amons is not presenting that one today, but he would be the person to chat more with about that. And he's in the front row, right below you.
Any other questions? Be right there, Cass. Uh-huh.
I appreciate that Mark is getting his steps in. So that was a fantastic talk, and as a non-expert, I really learned a lot, and that was fabulous. And I really appreciate the integration of your journey as a mentee to your journey as a mentor. And I just wanted to ask you what advice would you give the undergraduates and early graduate students in this room about how you go from I'm here by myself and I have no idea what to do to find a team to having a team? Like what recommendations do you have for like making those first contacts and helping build your own network?
Yeah, I think, well, really what I wanted to highlight was that at each stage that I've been in, someone helped me. But I think my selection of mentors didn't really have as much to do with the subject matter as it did with just people that I really enjoyed working with and trusted, and that dictated my career path, I think, to a large extent. And I think that's the main thing I would say to people when they're looking for the right mentor-mentee situation is emphasizing that rather than the subject matter that you want to study, and it'll work out. But I think there's generally, I think this department has always been that for me, if people are really willing to support and want to be mentors to people willing to work hard. So that's always been my experience here.
Maybe time for one more, if there is. All right. Be right there, Gilbert.
Thank you, Mark. Good job, Lynn. I really enjoyed it. So I'm a horse enthusiast, and so the kind of horses I have and I've grown up with in the community are not really considered purebred horses. So they're traditionally, probably don't have the ability to do what I'm looking at there on the screen. So, so they're I guess different breeds. I guess my question would be the results that you have obviously are associated with likely purebred horses and maybe performance in that way. Do you anticipate the data as it begins to, if you look at other animals like mustangs, for example, do you anticipate similar types of outcomes with your data?
Yeah, it's a great question. The human OA data has supported microbial differences, at least geographically and with diet. And so I would envision with those changes, at least, certainly the population you're talking about would probably have differences. I guess I should specify, these horses were all quarter horses and all similarly housed, essentially. So yes, I think it's something that warrants further investigation. We are enrolling microbial cases with a group in Kentucky too, just to at least have one comparison to go on. And then also with different types of OA, so post-traumatic versus longer-term degenerative joint disease, and then in obesity versus not. So I think all of those factors likely play a role. Certainly the main trends in humans with OA have just generally been an increase in firmicutes and an alteration in the firmicutes and Bacteroidetes ratios in OA. That has held true across the board, regardless of these other factors, but definitely there were a lot of other factors that affected it as well. Thanks.
Yep. Thank you.
Okay, thanks everyone for those great questions. I think we're going to move on to awarding the Zoetis Award to Lynn, which obviously is well-deserved. But can we have a round of applause for Lynn real quick? And I just have one quick announcement. As I said, the Undergraduate Research Symposium poster session is going to start immediately after the awards. If you are an undergraduate poster judge and have not signed in yet, you need your judge badge because your QR code is on the back, and that is how you access the judging form. So, yeah, so if you could go back over, you're going to get your steps in too. You have to go back to the Laurie Student Center where you check in, grab your judge's badge, and then come back over here and then check out your posters and your presenters, okay? With that, I'm going to invite Lisa Bergeron from Zoetis up to the stage to say a few words and present the Zoetis Award to Lynn. Lisa?
Hi, thanks everybody. I'm Lisa Bergeron. So I did need the microphone. I'm Lisa Bergeron. I'm a senior research director at Zoetis, and what a fantastic talk. I have so many ideas now about equine OA that I need to sort through in my head. But I'm also the site lead for our Colorado locations here in Fort Collins, just right on-site and off-site of CSU's campus. And just a real cheap plug real quick. I'm joined by some of my colleagues. We have a booth down the hall. Please come visit us. We're happy to talk about whatever is on your mind, even if it's not veterinary related. But right now, it's my privilege to be able to hand off the Zoetis Research Excellence Award, although I just heard that the name might have changed a little bit. But like Sue said, we've been giving this out, not me personally, but we've been giving this out for over 20 years to veterinary schools across the country, with the intent to recognize outstanding contributions towards veterinary health nationwide. And so the recipients of this award, as you saw, have fantastic research going on and are seen as not just contributing significantly to the field, but are also either they're on track to be nationally or internationally recognized for their work. And I really like the quote, if you want to go far, go together. So at Zoetis, our mission is to enhance or advance care for animals. And so we have to do that together, and that's why we're here. And so it's really a treasure to be able to hand out this award and celebrate the ongoing research in the field each year. So with that, congratulations and thanks for all the work that you do to make the rest of our work easier.